Good morning. My name is Devon Picklum, and today I'm going to be telling you about floral signals and the natural history of a Sierra Nevada wildflower system. So within this mixed meadow, you can see shooting stars, which are Primula tetrandra or Dodecatheon, and elephant's head, which is particular Scronelandiga. And here is Dr. Lazarus Macior, who conducted a lot of natural history research with these two species in the 1960s and 70s. So with these studies, he showed that these plants are wet meadow habitat specialists. They flower synchronously. They both require their bumblebees as pollinators. They provide only pollen and no nectar as a reward for these bumblebees. They also require a specific handling mechanism from these bumblebees in order for the bumblebees to access their pollen reward. So this is called floral sonication. And with this observation, he showed that there was multimodal floral trait similarity. So this is pretty special, and he thought that perhaps this floral similarity may increase plant reproduction in communities where these plants co-occur. And so with all of this amazing natural history work, there's still a handful of remaining natural history questions left unanswered. So in the first part of my talk, I will tackle some of those questions. And then the second part, I will use some new information we have about how bumblebee vision works, and in order to answer some questions about how similar the floral traits are actually for these plants. And so Macior's floral similarity facilitation hypothesis here, I like to think of the bees interacting directly with each of these flowers, driving this indirect interaction between these plants. So first off, I did a self-compatibility trial and demonstrated that pollination is required. So pollen needs to be moved by bumblebees between plants in order for reproduction to happen. Second, I observed bumblebee foraging and found that bees visited both species during a foraging bout, which tells me that the indirect interactions are happening in these meadows. And then I took some swabs of pollen off of bees' bodies. And so I did this by catching and cooling bees, which make them sleepy, swabbing them with fuchsin gel, and then letting the bees warm up and fly away. And so I took swabs from different parts of their body, their dorsal side and the ventral side of their abdomen, and looked for pollen under the microscope. When what I found was that on the ventral side, so that's underneath their belly, on their abdomen, there's a lot more shooting star pollen and a lot less elephant's head pollen. And on the dorsal side here, there's a lot more elephant's head pollen and a lot less shooting star pollen. And so what I'm taking away from this particular finding is that pollen is somewhat partitioned in where it is placed on the bee's body, but this partitioning is incomplete. And this really would allow for pollen transfer between these different species. And the next thing I did is I went in these same meadows and collected stigma samples. So these are the female reproductive parts of these plants, and looked for pollen. And so I found that with the shooting star stigmas, they almost exclusively had shooting star pollen, with a very few individuals having some of both pollen types. But with the elephant's head, while most of them still had elephants that had pollen, a substantial portion had pollen from both species. And then there were some that only had shooting star pollen on them, which I think is quite interesting. And that really, to me, might indicate an indirect interaction between these two species that is asymmetrical. And something further to point out is that, the elephant's head have a lot more stigmas per plant, so they may have more tolerance for the wrong type of pollen finding its way onto the stigmas. And the shooting stars have a lot higher population densities. And so that might be why you see more pollen from the elephant's head, is just that there's more of it in the environment. And so to recap, I found that pollination is required. Bees visit both species during a foraging bout. Pollen is partitioned but incompletely, and heterospecific pollen is sometimes found on stigmas. And so this really gets at the indirect interaction portion. I can't yet say if they're actually facilitating each other, but I haven't yet tackled the similarity part. So that's my next part here is floral similarity. So I like to sort of step back and think about these plants from my own perspective. I don't think that if I were in a meadow, I would easily mistake these two for each other. They look pretty distinct from my eyes. However, the way that you and I perceive things is quite different from how a bee may perceive things. And so we know from lots of detailed lab-based studies a lot about how bumblebee vision works. And so first thing we know is that they actually have quite different visual acuity than we do because of their compound eyes. So visual acuity is basically the ability of an eye or a camera or something to resolve a picture. And so you can think of this as what is happening in the eyes and the visual processing portion of the bee as it sees a scene. And so here I have this model showing approximately what that scene would look like to a bumblebee, a 5 centimeter tall shooting star at about 15 centimeters is going to be very blurred out and hard to differentiate. And at 10 centimeters, it's a little bit more resolved, but it's really still very unresolved. At 5 centimeters, you can start to see individual petals and individual parts of this flower, but it isn't really until you get right up close at 2.5 centimeters away that you can really, the bumblebee would really be able to resolve that picture in a way that we can interpret. So that's pretty astounding to me and really gets at kind of maybe at these mid distances, those dramatic differences in shape that these plants have, they may not be super important. And so maybe at fine scales, they can tell the difference in shape and color and structure, but at further distances, it may be just the color that's important in how they're differentiating these plants. So that gets me to the second part here, which is spectral sensitivity. So we also know a lot about the color receptive part of how a bee views the world. So up top here is how humans see the world. So this is the spectra of light from really narrow wavelengths of light to broader wavelengths of light that we can see. So we see blue to red. Bumblebees can see smaller wavelengths of light than we can into the ultraviolet, but they don't see super well in the red range. And so here's an example of a lot. I went out in the meadow and I used a reflectometer to measure floral color. And so these are on the x-axis here, we have wavelengths of light, and on the y-axis is percent reflectance. So basically these flowers reflect a lot in the blue end of the spectrum and in the red end of the spectrum, but not at all in the UV and very little in the middle here. And so I'll point out, bees aren't going to see a whole lot here. So to me, these plants look fairly pink or purple. A bee may see them more as a blue color because they don't really see a whole lot in the purple or in the red. And so in order to actually quantify, how different these colors are to each other, I'm going to use a visual model. So I'll walk you through how this works. So I use these two different dodecathion here, or they're shooting stars. And I chose one that was quite dark in color and one that was quite light in color. And the dark one here, you can see the reflectance is a lot less, it's a lot less reflective than the light one is quite reflective. And so the color can show up in their reflectance. And we can use a visual model. So these are called hexagon plots to plot each of these colors in 3-dimensional space. So this is kind of a foreshortened 3-dimensional plot. And we use this 3-dimensional plot. So it has the blue axis here, ultraviolet and green axis. And we use this foreshortened plot because we can actually map how well a bee can see on this specific plot, because people have done a lot of research with these. And so using the colors I've measured at different populations, I can actually go through and plot each of these points on this hexagon plot. So here I'm going to zoom in because these are all really quite close together in color, because overall, both of these are quite close in color. And so here, we can measure the distance, the color distance in hexagon units. And so these are all the abstract, but basically 2 points that are closer in space are going to be closer in color, and further apart points are going to be further apart in color. And here I have the elephant's heads in red is, and with the population average in the middle here, that point, and the shooting star in blue here with the species average in the middle there. And so I'm going to set this plot up for you because there's a lot going on, but basically same deal. We have our distance in color, our color distance in hexagon units. So again, that's just how similar or not similar these colors are. And here I've added these lines. And so from behavioral studies, we know that above about 0.11 color hexagon units, these colors are easy for a bumblebee to discriminate. That means that they can tell them apart from each other, no problem. But below about 0.04, those colors are really difficult for a bumblebee to tell apart from each other. It just, there's not a lot they can do. That's the limit of their ability to discriminate. But within this boundary here, between these two lines, the discriminability of these colors is kind of dependent on what you're asking the bee. So in these studies, they often will, provide options for the bees in different colored things and say, can you tell them apart? And often what those are rewards. So like a sucrose reward, something nectar. And so when they are provided with really different rewards, this one's really sweet, this one doesn't have much sugar in it, then the bee is a little bit better at telling those two colors apart because it has a reason to really work at it. But with rewards that are quite similar, they don't really differentiate between them. So I think that's really interesting. It's kind of how important it is for the bee to really hone in on how different those colors are is dependent on what is being offered in terms of a reward. So what I've done here with these color plots is within this population here, I compared an individual color to that species color average. So I call this conspecific color here. So comparing an individual to its species population average. And I've done that for both species here. And you can see that overall the elephant's head they're very close in color to each other. All of the individuals within this population would be indiscriminable from the color average. And so the reason I use this color average is it's kind of like the search image that the bumblebee may have for, okay, I'm going into the meadow and I'm looking for some elephant's head, and this is what color it is. Same for the shooting stars. I'm going in the meadow, this is what I'm looking for. So this shooting stars overall have a wider range of colors, and you can see that in how discriminable those colors can be. I also use comparisons of individuals within one species to the other species color average. And so I do this because I want to know how easy would it be for an individual elephant's head to be discriminated from the color average of its shooting star neighbors. And so here you can see both of these groups are going to be difficult for a bumblebee to discriminate from each other, and there's variation. And so I did this for several populations. So here at Yuba Pass, you can see overall everything is quite similar in color. But here at Levitt Lake, you can see in the hexagon plot, those population level blobs are overlapping, but not completely. And it really shows up in this discriminability plot where you can see some individuals are quite different to each other. And it really shows up. Maybe those individuals will be easier for those bumblebees to tell apart from each other. And really what I'm getting at here is that there is strong color similarity both within and between species. However, there's some variation, and that variation differs by population. So this is several more populations I found, and just really looking at how wide those variation within those species and also how much they are overlapping with each other. And overall, the variance of the elephant's head typically fits within the variance of the shooting stars, which I think is really interesting. And generally, there's a broader variance for shooting stars than there is for elephant's head generally too. And so to recap, I answered some remaining questions about natural history in this system, and I looked at just how similar these plants may look to each other, at least with regards to their color, from using bumblebee visual models. So with that, I'd like to thank you for listening and thank my advisors and co-authors here and for their help and guidance, and a field assistant, Rebecca Gaxiola here. Thank you.